Hey everyone, welcome back to the EduHaven course on AP Biology. This is going to be a done with pencil video on genetics, part 2, the genetics unit for AP Bio. Um, and without further ado, I hope you succeed. Now, DNA, it's a very interesting molecule because for a long time we didn't know what was the I think it's called the transforming principle. Yeah, transforming principle, the transforming factor. We didn't know what DNA or what nucleic acids actually did, um, because we see like proteins. We know that proteins, there are like practically infinite number of ways to fold a protein, which means there's an infinite number of uses for proteins. But we didn't know, like we had to prove that DNA was storing information, storing genes, right? So there's three critical like experiments that you have to know, or they're interesting. These are like the history. So there's the first one is the Griffith, Avery, McLeod, and I think McCarthy's in there too. It's really a series of experiments because I think it's I don't know the history exactly, but it's like two of them did it together, then another two replicated it, or maybe they went further. But overall, this is what happened. So Griffith, Avery, McLeod, McCarthy. So they had three rats. I don't know how to draw rats. I'm gonna do this. Three rats, and they had a pneumonia bacteria, and it was pathogenic. It was harmful or foreign, right? So they had a little. Here's my little bacteria. They gave three kinds of bacteria to these rats. A little unethical, but it's science. It's the real history. So the first one was pathogenic. It was harmful. So they put it in, and it died. Yeah. The second one, it was non-pathogenic. It was not harmful. So this one we'll label it as S, says for safe. This one survived, right? And then this one was pathogenic. However, they boiled it. And so now this one was dead. And then they gave it to him. And this rat, stayed alive. Now, they took another rat and they were like, you know what? What if we gave him, this rat, uh, both the non-pathogenic bacteria and the dead pathogenic bacteria? And they took these two, so they gave them the S and they gave him the dead one, the dead pathogenic. And they gave it to him. Sorry, they gave it to him, and this one died. And this proved something, because so the way they knew this was so these were, they had like bumps on them. They were rough. And so what happened was this dead one, it was transferring its genes, its pathogenic genes into the non-pathogenic bacteria. So this non-pathogenic bacteria was carrying the pathogenic gene from a dead one, and therefore it meant that DNA was the one was like was or it was hmm, it was like they knew that something was going on. There's a deeper factor because this one, all the protein's gone, right? Because it got heated up. So that's the first experiment. So it it furthered the knowledge of DNA. The second experiment is called the Hershey Chase experiment, and it's not related to the candy. Um, so the second one is called the bacteriophage. Ooh, I'm not really good at drawing them. I think it's like, it looks like an alien, like sci-fi monster. It's like this thing. Now, the interesting thing about the bacteriophage is that it had both. So inside of it, there was a little bit of here. On the outside, it was made out of protein, and on the inside, it had DNA. That's my DNA. Not my DNA specifically, but that's how I draw it. And What these things do is that they inject into a bacteria. They inject something into the bacteria to kill it. And what scientists did was, so remember the radioactive isotopes from earlier? They pretty much labeled the protein and DNA. Now, it was pretty smart. So protein, we know that protein, they have sulfur. Cysteine has sulfur, the specific amino acid. So they labeled it with a certain isotope of sulfur, while DNA has phosphorus. So they labeled it with a certain radioactive isotope of phosphorus. And once they injected it, they only found the phosphorus isotope in here. And that meant that DNA was the transforming principle. Because if protein was a transforming principle, the DNA, like if the protein was here, it would be seen in here. And what was happening really with the how this thing kills bacteria is that it takes the DNA. This DNA is used this transforming principle, I'll call it, is used to create proteins, and these proteins then kill, or they're used to create more bacteriophages inside, and those bacteriophages kill the bacteria. And so therefore, because DNA, only the phosphorus was found inside, DNA had to be transforming principle. That's experiment 2, Hershey Chase. And the last one is called, I don't know, chronologically it might not be the last one, but it's the Watson Crick slash, what's her name? Let me look at it. The Franklin, Rosalind Franklin. But we'll get into that in a second. So Watson and Chase, or Watson and Crick, not Chase, they were scientists. So Watson and Crick, they were, and Rosalind Franklin too, they were taking DNA and they were shooting X-ray at it, and they were documenting what it looked like. And what they found was that DNA had this weird pattern to it. And they're like, what? Why does it look like that? It was actually, I think it was more like that. But Watson and Crick figured out that DNA was a double helix. It was a double helix. Sorry, I'm not good at drawing, but they found that it's a double helix because imagine if this thing was like 3D. So it's like that way. The things line up like this. So that's why actually from the top down, it looks like an X. Because when they shot the X-rays at it, because it was a double helix. So that was interesting. And what I found interesting about it is Rosalind Franklin was a female scientist. She worked on it too, but the thing is, she died because she was working with X-ray. But she figured out that it was a double helix, but she doesn't really get credit for it. But that's a little bit of history. So, woo. So the next topic is mutations. Now, mutations occur in two main ways: with DNA polymerase errors, and we have mutagens like nicotine. They can change like bases. Then we have UV rays, which can form when they hit DNA, they can form thymine dimers, which can change the way it's read. And we have ionizing rays, which will hit chemicals in cells that ionize them into free radicals, and the free radicals are baneful or are harmful to DNA. And those are the main ways that DNA can be mutated. Now, the thing about DNA is that DNA polymerase kind of proofreads, so most of them can be fixed. And also, most like mutations, because of the way we have 64 codons leading to 20 amino acids, we have redundancy, which means that most mutations aren't actually leading to anything. Now, there's two types of mutations. There's a point mutation. So when you have a point mutation, it changes one base, or it changes one point. This leads to, this marker is messed up. It's unfortunate. This leads to a single base being changed. This means that there's like one single codon. So let's say this is the original. This will be the new one. And this single, the missing space is the chain. This is where the mutation occurred. And what this means is, what you have to remember is that, remember the central dogma. The change in the DNA will lead to a change in mRNA. A change in the mRNA will lead to a change in the polypeptide. And change in the polypeptide, and the change of the shape of the polypeptide based on the base or the amino acid, will lead to a new process being made. And this is critical because now this DNA will lead to a different mRNA. This mRNA will lead to different amino acids, potentially. Now, so that means if the original protein shape is this new one could be like, could be a different shape altogether. It could be like that, thanks to the new like a mutation. However, there are three types of mutations that you need to know of. There's three types of point mutations specifically. You have a nonsense. A nonsense. A nonsense means that. One sec. Okay. So we have three types of point mutations possible. The first one is a silent one. This means that this new codon, thanks to the redundancy of the base to codons, or the redundancy of the codons to amino acids, leads to a similar or the same exact amino acid. A similar as in like it has the same properties. This means that the folding will be the same, and therefore the enzyme and the process that it runs is the same. So there's no change. However, the next one is called a missense. A missense means that this change in the codon will lead to a change in amino acid that's different, and now a new folding is going to occur in the amino, because of the different amino acid properties. A new folding of the protein occurs, and a new process occurs, or a different process occurs, or the wrong process occurs. They all mean the same thing. So what this means is that, and the last one is a nonsense, which means it's an early stop codon. So let's say, like, I don't know, maybe let's pretend UAG is one of the stop codons. UAG is accidentally put in, or accidentally coded for, by this mutation, by the point mutation. That means the original is like, pretend the original is like this. Now new one is the exact same. I probably shouldn't have made it this complex. Let's pretend it's like that. So it's the same exact thing at the start, but then it just stops because it's an early stop codon. Now, one good thing about mutations is actually they're actually beneficial in some way, because if a missense occurs, or a new protein is made, a new process is made, you can either get a new function entirely that's like a new trait. For example, we have opposable thumbs, right? This was originally a mutation, and excuse me, um, this was originally a mutation, and it was beneficial, and we'll get to that in the evolution part of this course. But pretty much, it passed on through generations because it's beneficial, and now we all have opposable thumbs. And also, a new way of doing a process could occur. For example, um, I know this obviously hasn't happened in a long time, but let's say a mutation occurs, and now you can run cell respiration instead of one glucose leading to 36 ATP. You can now create 50 ATP. Now you don't even have to eat as much because um, you can create more energy. You can release more energy from less food. Now, does that stop me from eating the same amount as I do? No. But we don't get into that. The next one is a frame shift. So there's two types of frame shift. They're called frame shift because the way the codons work, it's like 3, 3, 3. If one of them's removed, it changes. So it's like 1, 2, 3, 1, 2, 3. However, if this one's removed, it's now going to be 1, 2, 3, 1, 2, hypothetical 3. So now that's the way a frame shift works. Now there's two types of frame shifts: insertion and deletion. The insertion ones, like the name suggests, insert a base, and the deletion ones will delete a base. This leads to a very different protein because, so like everything from the start is the same. Let me go simpler. I really need to stop doing horribly complex. So let's pretend this is a protein. That. Now, the new one will be like, let's say there's a deletion over here. So we go up, and then there's a deletion. So now the protein is going to be like, let's say it's this brand new one. So it's like that. So now there's an obvious change. So that's what a frame shift can do. It's the same until the point, and then it will change in the protein. And remember, a change in the DNA will lead to a change in the mRNA. A change in the mRNA will lead to a potentially different, or a different codon will lead to a potentially different amino acid, and a different amino acid with a different property means that a different folding of the polypeptide, which means a different function. And those are the two types of mutations that can occur. Let me see if there's anything important. Okay. So the next topic is recombination. So the thing about so we'll get into this in the evolution unit, but species uh they need genetic differences. They need genetic differences in order to like survive as a species. Because if everyone's the same, if everyone has a harmful gene, they all die. If everyone had cancer in the world, we'd all die. So, um, or potentially all die. I don't want to be morbid. Um, but so there's multiple ways of introducing genetic variation. Of course, with both of them, we have mutations with both eukaryotic and prokaryotic. But along with that, we have with eukaryotes, we know we have meiosis. So we have like different and we have the process of meiosis with independent assortment, um, independent assortment. What's the other one? The Mendel's laws. I'm blanking right now. Here, I'll put it somewhere. I'll put it somewhere on the board if I figure it out. But we have the meiosis creates variation. Another way is that we have two parents, or so I try to keep it simple for you. We're gonna call it multiple parents, because then we have three M's, multiple parents, and this means that our genomes are then are the combination of two different species. Not two different species. Oh my God, that's wrong. Two different individuals. So now our genomes are all different. So that means that introduces another way with prokaryotes, or with these ones, they also have three types. So we have inversion. So within little chromosomes, that's a chromosome or a chromatid. within one, that's a horrible drawing. Within uh, one of these, this part can just come out and just flip over. So let's say we had positive, negative on the, these are just markers we're going to use for now. It's going to flip, and now it's negative, positive. So it's like it comes out, inverts, comes back in. That's called an inversion. Another one is in transposition. So with transpositions, they're like jumping genes. They cut or paste or copy and paste. They they take a gene, cuts or pastes, and gets pasted pretty much. It jumps around. Yeah, and those are the ways eukaryotes work. Now, the last one, I'm gonna write jumping genes, because these last two are a good segue because they are involved with prokaryotic genetic recombination as well. However, there are three main ways that prokaryotes will, or there's three main ways along with these two. Three main ways that they introduce genetic variation. The first one is conjugation. So the way I remember it is I take Spanish. I don't took Spanish. I take Spanish. Um, and so con means together, or con means with. So the way I remember it is con. It's called bacterial sex. So what happens is it's two bacteria form a pilus or a bridge. It's a pilus is the real name, but it's a bridge. And this bridge will they will transfer, they will copy plasmids or its DNA, and they'll transfer it. And this introduces variation. The next method is transformation. Now, this is a very important one for us, and we'll cover it more later. But the way it works is a plasmid from the environment. A plasmid is just a small ring of DNA that bacteria have. It will go into the bacteria will take in plasmids from the environment. That's what transformation is. A separate type one is called transduction. This is when a virus or something like a bacteriophage will insert DNA uh, will insert DNA into let me change the color. Will insert DNA into a bacteria, and this is another way that genetic variation is introduced into these species. And that's that. So now that we've gone over how genes are expressed, or how genes are expressed, let's go over epigenetics, or this is called gene expression really. So it's how genes are like when they're turned on and when they're turned off. There are six major ways they're controlled in eukaryotic cells, or yeah, six major ways. The first one is the most important. It's transcription factors, and let me briefly go over them now. Transcription factors. So remember, we have let's say we have this gene right here. Gene X. You know what? Let's go gene Y. We'll go gene Y this time. Why? Because I gotta. I'm just kidding. Because so then we have the promoter behind here. So the most common transcription factors is called a repressor protein. These repressors, they will attach to an operator, and these operators, they really can be here, they can be here, behind, in front, they can be even on top of it. What they do is the operator. So this is where the DNA or RNA polymerase is trying to be. However, the operator will attach a repressor protein. This repressor protein will act as like a little wall, or it's pretty much going to stop DNA polymerase from going. That looks weird. it will stop DNA polymerase from going. So you now cannot transcribe this gene, and therefore you cannot translate the gene, and therefore you can't express a trait. You can't you can't express a trait. Now, another way this happens in eukaryotic cells is called enhancers. So upstream from this gene, so let's say this is we're gonna say this is the gene right here. Upstream, you'll have these enhancers, or they're enhancers. You have multiple enhancers. So this is an enhancer over here. There's like it's just small snippets of DNA, and what these do is activator proteins will connect to them. So we'll have an activator here, an activator here, activator here. And now, I think I got a load of blue in the red. That's unfortunate. Now, these activators will curl the DNA. So now it's like this. It's like this. And now, let's say this is the gene over here, and this is the operator. Now, it essentially drags the RNA polymerase into position. So now it can freely go. That's another way they're controlled. The next 6 of them, they're kind of like they're not like that important for you to know the nitty-gritty details, but you just need to really know, like, their name kind of explains them. So the second one is methylation. So methylation, they're called chemical tags. Methyl is the if you take chemistry, it's like the actual or like molecule that attaches. So it's methylation of the cytosines on DNAs, and this will deactivate um the DNA, the DNA slash gene of cytosines. The next one is I kind of remember them in like a kind of a janky way. So the next one, so methylation of cytosines. The next one is RNAi, or even before RNAi, because I want to keep it in order, is tight coiling. So remember, when DNA is inside the nucleus, it's chromatin. It looks like a big salad or a big bowl of spaghetti. And when you look at it through like really deep, there's like proteins, and these wrap around. So that looks weird. So you have this protein here, and the DNA, we're going to make red DNA. The DNA will wrap around the protein, pretending it goes around the back. And when you tightly coil it, it prevents it from being read, and therefore you cannot transcribe it, and therefore you cannot express it. So that's another one, tight coiling of DNA. The next one is RNAi. So RNAi, I'm pretty sure it's inhibitor. It might be interference or something, but it pretty much it chops up the mRNA before it gets onto the ribosome. Therefore, if it's chopped up, you can't translate it. Therefore, you can't express it, or you can't turn into a polypeptide. The fifth one, similar to transcription factors, they're translation factors. In essence, it's just proteins that can control, not promoter, that can control the expression okay. And the last one I kind of went out of order here. The last one is the variation in splicing. So remember how after transcription in the nucleus, they splice out the introns in eukaryotes and nucleus. With prokaryotes, one difference that you need to know is that with prokaryotes, transcription and translation happen at the same time. So they don't like splice out introns. They don't do that because it happens at the same time. So variation splicing. So based on like the way you splice the introns, because you don't have to take all of them out, you can take some of them out. The variation can lead to different mRNAs being sent to being translated, and therefore different proteins being made. Therefore, different genes are different processes are run because of a different protein means a different process because of a different shape. So these are the 6 ways they occur in eukaryotic cells. In prokaryotic cells, what you have to realize about prokaryotes is they're single-celled, right? So they don't really have body systems like we do, or like eukaryotes. So they can't respond to environmental changes as quickly. This results in them needing to have operons. So with prokaryotes, they have several genes. We're going to call it gene A, gene B, and gene 3. Because you never have to let your opponent know the next move. So they have operons. So they of course have an operator. I think I just messed up. So I just flipped these. I'm gonna have to fix in post-production. It's called a promoter. I apologize for that. So this promoter, it works normal. Now, you know what? Let me fix the way it looks because I don't like it. We're gonna change the way it looks. So we're gonna have a promoter over here, and then we have an operator. Now this operator, it binds repressor protein, right? Now, there's 2 types of operons that you need to know. So let's say this is the repressor protein. We're going to go with the a trip operon first, a tryptophan operon. So you have trip and lac, or this is lactose and tryptophan. Lactose is like a food. So tryptophan, it's something they build. It's something cells build. So what happens is when they have an excess, sorry, if they have an excess of tryptophan, they don't need to build anymore because they already have too much. They don't need to build anymore. So what happens is the tryptophan will attach here, and now will attach to a repressor protein, and now it will. So let's say this original shape is a triangle, and we know that triangles can't go in squares. If you passed like kindergarten, or they might be teaching it earlier. But now it changes the shape of the protein. So now it's a square or a rectangle. Now this is able to plug in here because it's the same size, and therefore it's able to block the promoter. It's able to block the RNA polymerase from transcribing the genes. And these genes create tryptophan. So therefore, you don't if you have more than enough tryptophan, it attaches, changes the shape. The repressor attaches to the operator, and now RNA polymerase won't transcribe and make more, and lead to the more production of the tryptophan. Now, the next one, it's the lac. Lactose is like food, like milk, lactase. So I think that's actually what it makes, lactase. So these create the genes, they create lactase to digest it. So when there's too much, so let's say in the beginning, this repressor is already binding. However, when a cell has the lactose in the body, the lactose will attach here and will attach to the repressor. Now, it will change the shape of the little plug-in part to another shape. So now it can't fit. The protein can't fit. The repressor protein can't fit, and therefore RNA polymerase is able to transcribe the gene. So it's the opposite of trips, and they're like opposites, and that's how operons work. The final topic of the genetics unit is biotechnology or genetic engineering. Um, there are four main ones that you need to know, and there's one additional one, but it's not really needed because it's kind of new, and it's also might be unethical. Um, but so there's five types. So and they all kind of play along with each other, and but they all kind of like work together in each other, and they're all used in different like ways. We'll do restriction enzymes first. So the first one is restriction enzymes. So restriction enzymes, like the name suggests, they're enzymes, and they're naturally found in bacteria because they cut up DNA, and this is used against like bacteriophages and invaders, so that they can defend against like DNA injection. And so what happens is these restriction enzymes are unique because they will take they will cut up enzymes, or they will cut up DNA at specific points, at specific points that are predictable. These points are usually have twofold rotational symmetry, or they are what's the word, palindromic. So this means um, that they are the same forwards on top as they are backwards on the bottom. So an example would be like AACGT. That's not true. I'm sorry. AACTT. Because it's now on the bottom, it's going to be AA. Oh, okay. You can't have an odd number, obviously. Duh. AACGTT. So this would be, backwards would be, or this way, on the bottom would be TTGCAA. And if you can recognize it, it's the same. So AACGTT. It's twofold rotational symmetry. And so it will cut. So the way they cut DNA is it breaks the phosphodiester bonds on the strong and stable phosphate sugar backbone. And there's two way it cuts. There's two ways these enzymes will cut. They will either cut like a flat one, or they'll cut staggered. The staggered ones are the most important ones for us. So they'll cut like this. This results in the DNA having sticky ends because these hydrogen bonds are weak. So they want to connect back, but they're very weak, so they very easily come apart. So these sticky ends, we have two sticky ends. So now we have these. Now, what this means is if we have two different like genes or two different pieces of DNA, we can combine um, with ligase from DNA replication. We can combine two different genomes using the same restriction enzyme because they'll cut along the same exact pattern. So that means the restriction enzymes will cut, create sticky ends. Their sticky ends of two different genomes will come together because they're from the same restriction enzyme, as they produce the same twofold rotational symmetry, like the pattern, and they'll come together and they'll be ligated. What this is useful for is in a special process called bacterial transformation. Yeah. So let's say we are a pharmaceutical company. I know they're hotly debated right now, but a pharmaceutical company, and we're trying to create insulin for our patients. So, uh, bacterial transformation. So we're creating uh, insulin. You can't really create insulin synthetically, I think. I don't know if that could be true or wrong, but the way it works is you make a bacteria create insulin. The way that works is you take a bacteria and you put in an insulin gene using a plasmid. So what happens is, so let's say this is a plasmid. It needs 3 parts. The first part is the restriction enzyme where it will cut, right there. The next part is it needs the gene of origin or the gene of interest. This gene of interest is usually, so remember, it's gonna cut the restriction enzyme is gonna cut it. This is the, let's say this is the insulin gene. We know that the restriction enzyme will cut here and here. So that means it was going to create a sticky end right here. So that means you can paste this little part and put it right there if we know that the restriction enzyme is going to cut here and here, or just here. You don't need to. It's going to cut here. So now we can just paste it in using restriction enzymes and ligase. Now, after this part is done, you also need, so this process is so this process can be done. However, for a bacteria to take up this plasmid, it's kind of rare. Even if you use heat, heat will increase the chance it happens. However, the chance that it still happens is still low. It means the minority. This means that you need one more thing. You will need an antibiotic resistance. Excuse me. You need an antibiotic resistance gene. This means that if you put the bacteria in antibiotics, that only the ones that have taken up the plasmid and have the antibiotic resistance gene will survive. And this leads to, if you do this experiment in your school or at like a university, this will lead to like little spots like this. When you don't put the little resistance, it actually creates a lawn. It's called a lawn because it's like all of them survive. However, if only the ones with the resistance gene can survive because we put antibiotic resistance, it creates little clusters. So now another part of this is called gel electrophoresis and PCR. I'm gonna erase it. So gel electrophoresis is a technique. It's also called DNA fingerprinting. The idea is, so gel electrophoresis. So the idea is it's based on the fact that so a gel phoresis, I might misspell it. But a gel, it's the special material that microscopically it has like these like tiny holes. It's like a filter. And so what happens is you create little divots in here. Whoops. You create little divots. And over here, you give it a negative charge. And over here, you give it a positive charge. And what this leads to is that when you put DNA in these little holes or little pockets in the gel, the DNA will travel outwards. Now, this DNA does not move uniformly. The DNA will travel outwards because the phosphate group in DNA makes it negatively charged, which means it moves away from negative, goes towards the positive. But it doesn't move uniformly. When you take restriction enzymes and cut it up, or you can, it's usually done with restriction enzymes. The DNA, short bands of DNA will move farther because they're shorter and they can squeeze through the holes a little bit more than the larger bands. So what this leads to is you get little bands like this. So let's say these are 3 different types of DNA. So you get little bands like this. So what this means is that the shortest DNA bands end up towards the here. And you can also compare this with like a ladder. And the ladder will be like, all right, over here, this is 500 bases. Over here, this is 200 bases. And so this can be used at DNA fingerprinting because let's say this is like the father and this is like a child that you don't know who the dad is. This can't be the father because the DNA, the bands don't match up. At least half the bands should match up if it's the father because of the mom and dad. So that's another part. The last technology is called PCR. It's polymerase chain reactions. So remember how um, PCR. So what it does is they're trying to um, copy DNA, like million, like hundred. Like they're trying to create a ton of the same strips of DNA. So what happens is they take DNA, they put little DNA primers, so they know like the little areas that they want to copy, and then it will it will start warm, go hot, go warm, go cool, and then it does this little rotation. It looks like a sine line, but in essence, it will try to copy a bunch of the so it because now okay it so in essence, let me finish my thought. Um, in essence, it will try to copy a bunch of the DNA. Now, notice how it's really hot. This is really hot, like 80 degrees. Most enzymes will, like in your body, um, I think it's 80 degrees Celsius, which is like super hot. But most like enzymes, like in your body, those enzymes will um they'll start shaking and they'll all the weak bonds will break. It's gonna like get messed up, right? What this means is that um you need a special type of polymerase. It's DNA polymerase because it's DNA because they're replicating DNA. It uses Taq polymerase. Taq is a type of bacteria found in hot like I think it's like magma pools, and what this means is that it's able to resist the heat, and so there that's why the fifth one I'm not going to write on the board. It's called CRISPR. It's this new technology. It's used in genetic like engineering. I think it's a little unethical because there could be like harm in doing it, but I don't know. Take it up with Gattaca. And that is it on the EduHaven done with pencil on AP Biogenetics. Um, if you enjoyed, please consider subscribing. And check out another possible done with pencil uh, video. So, or you can check out maybe there's shorts now. Um, but thank you for watching, and I hope you succeed.